Today's speaker. I'm happy to welcome today Carolyn Edelstein, co-founder and executive director of OpenBiome, a nonprofit microbiome research institute. OpenBiome was a 2017 New England Innovation Awards winner, and they have a very novel thing that they do. I'm going to let Carolyn talk about it and all the things that have been going on since she presented to us a year ago. Please welcome Carolyn Edelstein.
Thank you, Susan. And it was very, very exciting for our group to be honored with the New England Business Association's 2017 Innovator Award. And so I'm really excited to be back with you this morning talking a little bit more about the work that we do. OpenBiome is a nonprofit stool bank. And the reason that we thought the world could use someone to be banking and researching stool is a story that starts for us in 2011 when a cousin of mine went into the hospital in New York for a routine surgery and came out with a C. diff infection. And I don't know how many of you are familiar with Clostridium difficile, but it's a really aggressive intestinal infection that causes diarrhea upwards of 20 times a day, intense pain and nausea, loss of appetite, fever, and it's pretty dangerous. And Johnny spent the next 18 months trying to beat C. diff with round after round of antibiotic, eventually over 7 rounds of vancomycin, which is a last-line antibiotic. And he certainly wasn't alone. About 500,000 Americans get C. diff every year. It's the most common hospital-acquired infection in the country, and about 30,000 people die. And 1 in 5, like my cousin, don't respond to a first course of antibiotics, and they end up getting these recurrent infections where every time they go off antibiotics, C. diff comes back. And so after about a year of this, he started to look into other options, and he came across some of the medical literature talking about a treatment called a fecal transplant. And FMT, or fecal transplantation, is exactly what it sounds like. It's a sample of stool taken from a healthy person and infused into the colon of the patient, usually through a colonoscopic administration or a nasoenteric tube. The bacteria in that stool infusion outcompete the C. diff, and about 80% of the time, after one treatment, the infection is eliminated. And so Johnny thought this sounded like a pretty decent option for him. He wanted to find a clinician who could offer the treatment. He was in New York, in the healthcare space at the time, extremely well-connected and well-resourced, and if anyone was going to find access to this thing, it was him. But what he found was that there was only one clinician in New York performing fecal transplantation at the time. That clinician's wait list was about 6 months long, and at that point, Johnny honestly didn't think he could hold out. And so he ended up doing what a lot of patients were doing at the time, which was he went out, to the drugstore, got an at-home enema kit, talked to his roommate to ask him to give him a sample of stool, and ended up treating himself at home in the bathroom. And it worked. The next day, after 18 months of this, he had his life back. And so we were looking at this thinking this, cannot be how we're solving this problem. On the one hand, you have the most common hospital-acquired infection in the country, and on the other, you have a treatment that is demonstrating in the literature to be about 80-90% effective. And the only thing at the time bringing them together was an at-home enema kit and a very understanding roommate. We ended up looking a little bit into some of the barriers to try and understand why clinicians weren't offering this treatment. It was known for decades as sort of a fringe therapy for C. diff, but C. diff over the last, I think starting in 2010, a new hypervirulent strain had emerged, so C. diff became a bigger problem. The procedure itself was something that was very poorly understood, but was increasingly better understood because we suddenly had the tools to be able to characterize the bacteria that were living in our guts and to start to question, why this was working. And so there was a surge of clinical research interest in fecal transplantation. And those two things were starting to mean that at conferences you were starting to hear about FMT. But in practice, there were a ton of barriers. Clinicians, if they were going to provide this treatment to a patient, had to work with that patient to find and screen a healthy donor. That was expensive and very difficult. They also had to line up all the permissions in their hospital to be able to collect the stool, hope that the donor could perform on command the day of the procedure, work with that material in their endoscopy suites, and finally deliver it to their patients. And so, because of this, there were very few clinicians who were offering it at the time. And so we thought, my co-founder, Mark Smith, was working on his PhD at MIT at the time on research that was demonstrating that frozen and fresh stool were equally efficacious for treating C. diff. And from that idea, quite naturally came the concept of a stool bank, like a blood bank, so that patients like Johnny could have safe access and easy and reliable access to material that could be used in fecal transplantation. So we launched in 2013, really to just, reduce those barriers to access for clinicians, by extension their patients, by taking on the bulk of the work. And so instead of having to source your own donor, which would be crazy in the context of a blood donation, similarly here, we go out and we source donors, and about 3% of people make it through our screening process and can actually donate stool. And then we process the material in a manufacturing facility at a much higher level of quality than would be possible at a facility level. And then we send that material out frozen on dry ice for next day delivery around the country. And so, this model has afforded a lot of rigor and standardization to the practice. It's also offered economies of scale that has definitely reduced the cost of the procedure for patients and their facilities. And I think the other thing that was important that we could offer was, this practice was relatively new, at scale in the field. And by centralizing practice, we could learn from across the tens of thousands of treatments that we'd facilitate and the thousands of hospitals and clinics that we'd work with to really help with lesson learning and ensuring best practice across the country. And so in 2013, when we started, about 6% of the U.S. was an hour drive from one of the hospitals that we were working with to provide FMT, and we sent out 6 treatments that year. And I think the second patient that we treated had to fly from the Upper Peninsula of Michigan to Los Angeles to get her treatment, which is not ideal when you have chronic diarrhea. And so we really wanted to see that change. We wanted to make it, we wanted to see the number of treatments that we were sending out change and cover a meaningful fraction of the 40,000 patients a year that were at the time, failing all care and would qualify for a fecal transplant. And we also wanted to make it a lot easier for them to reach a doctor who could do this for them. So in 2014, we aimed to send out 1,000 treatments. We ended up sending out about 1,800 treatments, and half the population was within one of our partners. By 2015, that number rose again, and 2016, until by the end of 2017, about 90% of the U.S. now lives within an hour drive of one of the hospitals or clinics that we serve. And so all told, as of today, we've sent out about 36,000 treatments just for C. diff across a network of about 1,100 hospitals and clinics around the U.S. and a few overseas. Alaska and Hawaii are included in this number. It's just not as easy to put them on the slide. But one thing that we're very proud of is this expanded access to care. And broadly in the field, I think this has become, a really accepted practice for those patients who have failed multiple rounds of antibiotics for C. diff. Medical professional societies, the Infectious Disease Society of America and the American Gastroenterological Association have changed their guidelines, their clinical guidelines, to advocate for fecal transplantation being standard of care for these patients. And the American Gastro Association has also started a new project where they'll be enrolling 4,000 FMT recipients into a national registry, and they'll track those patients for 10 years. So we're really excited about all those developments. The other half of what we do, though, is, there's beyond C. diff, clinical researchers are really interested in understanding how the trillions of bacteria that live in our guts are interacting with our bodies and affecting our health. And in particular, there's been, I think, a groundswell of research drawing associations between bacteria and diseases from other infectious diseases, autoimmune disease, metabolic disease and obesity, and even neuropsychiatric disease like anxiety and depression and autism. And we don't yet understand whether or not these relationships are causal. We don't understand the nature of these relationships, but one of the first ways that we can start to understand whether or not we can use bacteria and manipulate bacteria to improve health outcomes is by starting with a fecal transplant. And the clinical researchers who are predominantly academic investigators who want to start that early stage research encounter all the same barriers that a clinician wanting to offer it to their C. diff patient would offer or would encounter. And so we've worked with across about 32 different studies in a broad spectrum of disease indications to start that early stage research that can hopefully point to new signals in other areas where some sort of intervention with the microbiome may lead to improved health outcomes. We ourselves also sponsor research, and so we've done a few trials looking at other antibiotic-resistant bacteria that could be outcompeted by C. diff. And most recently, in September, we're preparing to launch a trial at the University of Cape Town in South Africa looking at severe acute malnutrition, and I'll talk about that in a second. So we support a meaningful fraction of FMT research worldwide, and again, I think one of the key pieces of the key services that we offer is not just making it a lot easier for this research to happen, which is important, but we can also help aggregate, by providing a level of standardization to that research, and by sitting in the middle of it, we can help sort of aggregate from across all of these studies and drive up the N and see patterns that might not be possible if all of these were little disparate efforts happening without some sort of centralization. All that being said, I think we're very proud of what we've accomplished in the last 5 years, but in terms of, achieving safe access to patients, especially with C. diff, there are still significant barriers. Fecal transplantation is an investigational product. And what that means is FDA, as well as the medical societies I mentioned, have accepted it as standard of care as a last resort treatment. And so to get access to it, patients have to wait on average 10 months of trying and failing antibiotics before they're able to get a fecal transplant. And 10 months with C. diff is not something that I would wish on anyone. As another barrier, because it's investigational, oftentimes it's very hard for patients to get this covered by insurers. And so a lot of patients end up paying out of pocket, and we do have a small pro bono fund that we can support treatments with, but we don't always hear about folks who need that. And then there are also, I think, still, yesterday we fielded questions from, a major national network that has institutional barriers against being able to offer investigational products, and so none of their patients are able to get FMT. And so there's still a lot of, I think, problems with with achieving access to this product, and really the only solution is to see it gain FDA approval. And so in 2017, we spun out a company, and this is getting a little bit to our structure, but OpenBiome is a nonprofit, and pursuing FDA approval as a nonprofit is just not something we thought we could capitalize around, or that we should use, philanthropic resources to do something that the private market could fund. And so we spun out Finch, and Finch Therapeutics, we've licensed over our manufacturing program to Finch, and they now run it, and they've been able to improve its quality environment as well, and they provide the material back to us that we distribute for our C. diff patients and for the trials that we support. Meanwhile, Finch is pursuing FDA approval. They're in phase 2 trials right now for a formulation of fecal transplantation that'll be encapsulated. It's also lyophilized, so it's freeze-dried. It's a lot easier. It saves you on the procedure. It has a lot of advantages, but most importantly, I think it'll ensure that patients will have perpetual access to this treatment if it's successful. Also, if it's successful, OpenBiome is entitled to a royalty stream from the sales of that product that we will then be able to use to further the research that we want to do. And so when we look to the future, this is where we're particularly excited, is now we have this structure where Finch is pursuing commercial applications, and there's a lot of activity, actually a lot of commercial activity in this space interested in harnessing the microbiome to improve public health. But we see gaps that we think as a nonprofit we're particularly well suited to fill. And in particular, those gaps are in the long-term research into areas like preventing these diseases in the first place. And the area where we're focusing most immediately is in those microbiome-mediated diseases that predominantly affect patients in low and middle-income countries, and so are not going to be the first applications that commercial groups are going to be able to pursue. So our first trial in this area is going to be in malnutrition, and it's one of many potentially microbiome-mediated diseases that mostly affect patients in poorer countries. And one thing that we noticed was that there's a serious dearth of activity at all levels looking at patients, at patient populations in these areas. All of our observational data right now on just kind of collecting samples on the microbiome comes out of mostly wealthy countries as well. There are now at least a couple hundred FMT trials happening worldwide, but the only one that's happened at all on the continent of Africa is going to be ours starting in September. And yet there's a lot of really compelling preclinical data, and we're especially excited about some of the malnutrition work that Jeff Gordon's lab has done. So there's a fraction of kids who get severe acute malnutrition who don't respond to standard nutritional therapies. It's about 6 to 7 million kids a year who pediatricians struggle to treat because they don't seem to respond when you feed them, and they go on a protocol of antibiotics and then use of ready-to-use nutritional foods, and they don't seem to recover the same way most kids do. Jeff Gordon at the University of Washington ended up doing really interesting research in Malawian twins, where one twin was healthy and the other twin was malnourished. And he took samples of stool from each of those twins, fed them to germ-free mice, and the mouse that received stool from the healthy twin ended up with healthy growth. The mouse that received the stool from the malnourished twin ended up with all the hallmarks of severe acute malnutrition. And then what was really exciting for us was these mice were then co-housed, and mice are coprophagic, so they eat each other's poop, which is a really nice kind of FMT-style intervention. And in that case, the mice that had been malnourished had been fed the malnourished stool, ate the healthy mouse poop, and recovered, and showed, they were back on a healthy growth trajectory. And so for us, that's enough justification to start a small pilot at the University of Cape Town, and we have a pediatric gastroenterologist who's been a fellow with us for a couple of years, who we've been working on this protocol with. He's going to go back and start that study in the fall in a small, just 20-patient randomized trial, where primarily we're just looking to see, is this a safe intervention in this population? Is it feasible? And then we'll start to look at some of the biomarkers associated with severe acute malnutrition to see if we notice any signal there. So this is some of the super early stage research that we want to do, and the idea is to de-risk these interventions enough to be able to hand them off for further development. So beyond malnutrition, similar stories in typhoid, where there's been interesting case series data. Typhoid is caused by a bacteria called Salmonella typhi, and at least in a case series with some Finnish chefs who were colonized by Salmonella and so were kept out of work, and FMT intervention, outcompeted the Salmonella. They were decolonized. They were able to return to work. So we're interested in whether or not a similar intervention could be used for typhoid carriers. In stunting, there's also some early kind of murine model preclinical data to suggest that when you feed stool to mice, that they also, show some of the hallmarks of stunting. We haven't tried to reverse it yet, so there's more work to be done in that area, but another one that we're really eager to pursue. And so I'm just going to end on this slide. I think one of the things that is sort of interesting about our story is that, we are a nonprofit. We started with a small seed grant of about $125,000, and with that, we're very quickly able to start, recovering both that investment, but also then to grow with sales. And so, very much over the last few years, we've grown with sale of the material that we provided for C. diff, and we've been able to use those sales to also support research happening across the field and subsidize the work that we do with those partners. But what we're really excited about is, the yellow boxes sitting on top of 2017 and 2018 through partnerships like the one that we've done with Finch, but also with other groups that have started to return some resources back to us, and that we can then kind of use to endow some of the research that we want to do in the future. And so, we're interested in seeing if we can repeat the model the experience that we had with C. diff in other indications. Thanks. Yeah, I'm excited to take some questions and chat a little bit more about fecal transplants. Thank you. Yeah, it's a really good question. And we've seen for C. diff that pretty much any healthy person will work. So what we define as healthy is extremely rigorous. Because we don't understand these associations between bacteria and all kinds of aspects of health, we tend to exclude anyone who has anything with any association with the microbiome. But when we find those healthy people, what we've seen and what the literature has shown is that anyone works for C. diff. The same is likely not true for other diseases. And so there's been a lot of interest in using a fecal transplant-like intervention in ulcerative colitis, and there the early experience with fecal transplantation has shown that some donors are much more effective than others. Yeah, so in terms of commercial development, not us. So Finch has several programs that they're interested in, and Finch's approach is, similar to ours in philosophy, unsurprising because we're founded by the same people. But starting with that first experience in fecal transplantation, they're working to reverse engineer which bacteria are driving good, therapeutic outcomes, and then trying to develop those sort of rationally selected consortia of bacteria for other diseases beyond C. diff. So for C. diff, it seems like that full spectrum, all of the bacteria from poop seems to be a good product, but then there may be other places where either because the patient population is so large or because patients might need maintenance therapy, the idea of using material from donors is daunting. And so instead what they're trying to do is develop synthetic consortia of bacteria that will work. OpenBiome, meanwhile, is trying to encourage the development of treatments across all of these disease indications that we're interested in particular by supporting those early stage clinical interventions that can start to point the way towards potential, other therapies. Yeah, it would be very exciting. I think for our C. diff program over the last couple of years, we've done some work overseas. The regulatory environment is a little bit piecemeal, and there are most places. Part of what's unusual about this story in the United States is that FDA looked at this in 2013, and despite regulating it like an investigational drug, said, which ordinarily means that you'd only be able to offer it to patients as part of a clinical trial registered with the agency, they said, okay, clearly, for C. diff patients, the potential benefits outweigh the potential risks, and we're going to exercise enforcement discretion, and you can go and treat patients with C. diff who have failed to respond to standard therapies outside of a clinical trial. And that's how we've been able to serve such a big network so quickly, but the same policies, are only there in a few places, Australia, Ireland, but beyond C. diff, I think, and in particular being able to support clinical research overseas is something that we're increasingly interested in being able to do.